Welcome to the future of understanding. This is NeuroMind Insights, where we unlock the power of your mind.
Every thought, reflex, and sensation you experience relies on a single biological mechanism, the electrical firing of neurons. The brain operates as a biological computer, and this electrical spike is the language it uses to process the world. But an individual neuron does not naturally communicate in clean digital signals. It receives a chaotic mess of analog chemical inputs from thousands of neighbors. Its primary task is to synthesize that chemical noise and convert it into one binary electrical output. The foundational metric for this entire computation is membrane voltage, the difference in electrical charge between the inside and the outside of the cell. This chart maps voltage over time, tracing the action potential, the universal roadmap for a neuron's electrical spike. This entire wave is generated at the axon hillock, the funnel-like structure connecting the cell body to its long axon. To decode the language of the nervous system, we must examine the molecular mechanics happening at four critical voltage coordinates: -70, -55, +30, and -90 millivolts. The action potential is a physical timeline. It represents specialized proteins shifting shape in the cell membrane to propel a signal forward. We start at the baseline voltage on our graph, -70 millivolts. At rest, the inside of the neuron holds a negative charge compared to the extracellular fluid outside. The primary engine running the system is the sodium-potassium ATPase pump. It burns ATP to continuously push 3 positively charged sodium ions out of the cell, while pulling 2 potassium ions back in, creating steep concentration gradients. Ions naturally move from high concentration to low concentration. We calculate this DROI using the Nernst potential. The exact theoretical voltage where an ion's chemical drive to diffuse is perfectly balanced by its electrostatic drive to stay. If the cell membrane only allowed potassium to move, positively charged potassium would leak out of the cell down its concentration gradient until the internal voltage reached exactly -90 millivolts. However, the membrane contains leaky channels. The neuron is significantly more permeable to potassium than sodium. Potassium pours out of the cell continuously, while only a slight trickle of sodium leaks back in. Because of this uneven permeability, the voltage doesn't drop to potassium's theoretical Nernst potential of -90. The slow influx of sodium pulls the number higher, settling at a weighted average between the two ions. That weighted average lands at our starting coordinate, -70 millivolts. The resting membrane potential is a state of highly maintained electrochemical tension. It operates like a coiled spring, burning ATP to prepare for a signal. The axon hillock holds this tension while the rest of the cell listens. On the neuron's dendrites, the cell receives incoming chemical messages from neighboring neurons. These chemical messages interface with ligand-gated ion channels. These channels remain closed until a neurotransmitter binds to a receptor pocket on the protein, triggering a conformational shift that allows ions to flow. When a stimulatory neurotransmitter like glutamate binds, it opens sodium channels. Positive sodium rushes in, moving the cell's internal voltage up toward zero. We call this an excitatory postsynaptic potential, or EPSP. Conversely, an inhibitory neurotransmitter like GABA opens chloride channels. Negative chloride flows in, dragging the internal voltage downward toward -90 millivolts. This is an inhibitory postsynaptic potential, or IPSP. Looking back at our master graph, you can see these tiny fluctuations in the baseline voltage. The cell is experiencing a constant mathematical tug-of-war between excitatory and inhibitory signals. A single EPSP only shifts the voltage by a fraction of a millivolt. It is too weak to trigger a neuron to fire on its own. To generate a meaningful voltage change, the neuron relies on summation, adding these excitatory signals together. Temporal-summation occurs when one synapse fires repeatedly in rapid succession, stacking the voltage higher.
Spatial summation occurs when multiple synapses fire simultaneously. The target for all this summation is the critical threshold coordinate, -55 millivolts. The neuron acts as an accounting department. It mathematically weighs opposing analog signals until it crosses the -55 millivolt threshold required to make a digital decision. Once the internal voltage reaches exactly -55 millivolts at the axon hillock, the analog computation terminates. The system goes digital. This switch relies on the voltage-gated sodium channel. Hitting exactly -55 millivolts forces a mechanical change in the protein, instantly snapping open its outer activation gate. Driven by their gradient and the internal negative charge, sodium ions rush into the cell en masse. As positive sodium floods the interior, the internal voltage flips rapidly from a negative state to a heavily positive state. This is depolarization. On the grass, it maps as a near vertical climb, shooting upward from -55 millivolts and stopping at +30. Hitting the -55 millivolt threshold is a mechanical point of no return. It instantly converts accumulated chemical tension into an unstoppable electrical spike. But this voltage spike stops abruptly at +30 millivolts rather than climbing indefinitely. When the internal voltage hits +30 millivolts, a ball and chain inactivation gate on the inside of the sodium channel swings up and plugs the pore. Simultaneously, slower voltage-gated potassium channels finally open, and positively charged potassium rushes out of the cell. As positive ions leave, the cell interior rapidly drops back down toward negative territory. This phase is called repolarization. On our graph, the line plummets from the peak back toward the baseline. These voltage-gated potassium channels are slow to close. They allow too much potassium to escape, overshooting the baseline and driving the voltage to -90 millivolts. This is hyperpolarization. During the drop from +30 back to rest, the cell is in the absolute refractory period. Because the sodium channel is still physically plugged, it is impossible for the cell to fire a new signal, regardless of the stimulus strength. Once the voltage dips to -90 millivolts, the gates reset, entering the relative refractory period. The cell can fire again, but it requires a massive stimulus to bridge the 35 millivolt gap back to the -55 threshold. Behind all of this, the sodium-potassium pumps work to clean up the ion distribution, gradually pulling the voltage back to the -70 millivolt baseline to complete the cycle. The delayed opening and closing of these microscopic protein gates is what establishes a neuron's maximum firing speed. By locking the sodium channels temporarily, the cell ensures the electrical signal never travels backward. The action potential we just derived is entirely localized to one tiny patch of membrane at the axon hillock. But the initial flood of sodium diffuses laterally down the inside of the axon, bringing the adjacent patch of membrane to -55 millivolts. This triggers its neighbors, like a row of falling dominoes. This depolarizing wave travels the entire length of the axon until it reaches the end of the line, the axon terminal. The axon terminal is lined with voltage-gated calcium channels that open when the +30 millivolt wave arrives. Calcium floods in and binds to snare proteins on synaptic vesicles. This fuses them with the membrane, dumping neurotransmitters into the synaptic cleft. Once the terminal repolarizes back to -90 millivolts, the calcium channels close, cutting off the neurotransmitter release. The loop is complete. The cell maintained its resting tension, calculated inputs to hit a threshold, fired a digital electrical spike, and released chemicals to another cell. The entire derivation of the action potential serves one purpose: to bridge the physical gap of the synapse. This is the mechanical foundation that allows independent cells to function as an integrated biological circuit.
Thank you for watching. If you enjoyed this video, please like, comment, share, and subscribe to Neuromind Insights for more.